Thank you all for coming tonight. My name is Evan Chin, and over the summer I worked at a lab at UC Berkeley called the Nrf2 lab under my mentor HIf1a. And so the work of our lab in general focuses on various types of drug delivery and how we can optimize these systems. And in particular, my project is designing lipid nanoparticles for the co-delivery of Nrf2 and HIf1a in treatment mRNA. So for a brief overview of our presentation, we'll start with a background on lipid nanoparticles and how they can be used to treat diseases, go into our method for building the Nrf2 and HIf1a, then the results of this and our intended future pathways for experiments. Okay. So this is a concept that you might be familiar with if you've taken a high school biology class, but the central dogma is a way that we represent how living organisms store and make use of information. And so genetic data is stored in the form of letters in DNA, which is then transcribed to RNA, which is a type of intermediary, and then translated to protein. Now proteins end up being what do a lot of the things in your body. For example, many types of hormones are proteins, and also they're responsible for catalyzing many reactions. And so, logically, many diseases are associated with either having dysfunctional proteins or if you're missing proteins. So when we look to treat diseases, one place we often look is how can we get proteins into the body or into the cell. And how we specifically approach this in this project, at least, is that if we can deliver RNA into the cell, then the body can make use of this RNA, turn it into protein, which supplements what we need it to. And so the platform we are using to deliver RNA into the cell is called lipid nanoparticles. And so these are built of lipids, which if you remember from Lulu's presentation, are similar to the material that makes up the cell membrane. And what's important here is that they're capable of packaging mRNA. And so how these are used is that we inject them into a body, and then they get to a cell, get absorbed by that cell, at which point that lipid layer breaks down and the mRNA is released into the cell, at which point the ribosomes in the cell can make use of that mRNA and turn it into protein. Now, these have already been demonstrated to be very effective in the treatment of many diseases. There are LNP-related therapeutics for HIV, COVID-19, and cancer that have all proved to be effective. For example, one that you may have heard of recently, the Pfizer COVID vaccine is actually a lipid nanoparticle-based vaccine. And so one key detail here that will come up later is that lipid nanoparticle treatments often require multiple injections. Now, although lipid nanoparticles are proven to be revolutionary in the field of medicine, they do face some roadblocks, one of which is that they often trigger an immune response, which attacks the lipid nanoparticles and reduces its ability to get into the cell. And so, if you look at this diagram, this is especially an issue on later injections. As you see here, in the first injection, we see a pretty high effectiveness of our drug, but on subsequent injections, because the human immune system actually adapts to stimuli, we actually see on our later injections that this becomes less effective. Ideally, what we want to see is something more like this, where on these subsequent injections, we still have a high efficacy of our drug. And the genes we are using to try to accomplish this are HIf1a and Nrf2. These two genes are both transcription factors that are associated with low oxygen environments. And previous studies have shown that the activation of these two genes leads to a reduced inflammatory response. So overall, our strategy, if we can deliver the mRNA for these two genes alongside the mRNA we use to treat the disease we're trying to treat, we can potentially treat the disease and simultaneously lower that immune response. Now, the current phase of this project we're in is still kind of building these lipid nanoparticles, and so I'll get a bit into the process of that. To start, we take the DNA form of these two genes, HIf1a and Nrf2, insert them into circular pieces of DNA called plasmids. Then we check to see which insertions were successful. Then we can transcribe them outside of the body to turn them into mRNA, at which point we can package them into lipid nanoparticles and they can be injected and tested. Now, most of my work over the summer is on this process of screening, so I'll go a bit more into detail about that. So the reason we have to screen is that when we insert the HIf1a or the Nrf2 genes into a plasmid, it doesn't have the highest success rate, so we have to check to see which ones are successful so that we can guarantee that we're actually creating the mRNA for those two genes. And how we do this, we use kind of bacteria as a tool for this. Bacteria has a property where it actually takes up plasmids inside of it. And so once we add the plasmids to the bacteria, we will have some bacteria that have the plasmids with the insert, and then some bacteria with the plasmids without the insert. And how we distinguish between these, if you remember from Rita's presentation, is a process called PCR. Now PCR makes many copies of a specific sequence of DNA. This diagram, between these two black lines, PCR will make a bunch of copies of that sequence right there. And the trick we're using here is that when we have our insertion, that sequence gets longer because we've inserted a whole gene into that section. And then what that allows us to do is sort these by length. So we can then sort them by length through gel electrophoresis. How gel electrophoresis works is that, so it works on the principle that DNA is actually negatively charged. So when we put DNA into a gel, we can run a current through it, and so that DNA will be repelled by the negative electrode and pushed towards the positive electrode. The thing about the gel is that larger DNA gets stuck in that gel, so it will actually move through the gel slower. Now, as a result, the larger gels will be near the top of the gel, while the smaller DNAs will be near the bottom of the gel. And so just from some results from gels we did, this was an attempt to add HIf1a insertion, and I say attempt because none of these worked. But so how we do this, as I mentioned earlier, the longer sequences are at the top and the shorter ones at the bottom. And I labeled this blue box where we expect the failed insertion to appear on this gel. And so as you can see, these white lines here indicate that we have DNA there. On the other hand, in this box, that indicates with insertions, we have nothing there, which means no successful insertions. Now this is a gel where we inserted Nrf2 into plasmids. Here we actually do see a bit more success. And if you see this with insert box, we have some white bands, which means that these were successful insertions. However, these are not actually pure samples. There seems to be some contamination because in each of the lanes where we have the successful insertion, we also have a unsuccessful insertion in that same lane, which means that in each of these samples we have both a successful with insertion and a no insertion plasmid. So basically, in summary, if we try to transcribe these from the mRNA, we would get both the gene we want here, but also some RNA that we can't use for the nanoparticles. And so the main future pathway here, really, once we finish building these nanoparticles, is to be able to test them in living organisms in vivo. And so the model organism for this will likely be either mice or rats, as there tend to be a somewhat common model organism for experiments. And we would like to test for inflammation immune response to see if, when we add HIf1a or Nrf2, if that immune response gets reduced as we want it to. Also, the use of the treatment mRNA, if the treatment mRNA gets used in the cell, and also the ability of this, like in a broad scope, the ability of this platform to treat diseases when we need it to. And overall, I would like to thank my mentor, Kisong, and the Breathy Lab for all the guidance they gave me over the summer, and of course, the STEM cohort and Cameron.